So still we have 2 minutes. However, to save the time for the speakers, let me just start the session. Thank you for coming back. And this session is called Nanodrug Delivery, Novel Insights and Strategies. And the presentation will be 12 minutes, and the question and answers are 3 minutes. And after the entire presentation, we have 20 minutes more. So we can just relax. And the first speaker is Professor Dr. Wolfgang J. Parak from Philipps University of Marburg. And his title is What Happens to Nanoparticles After They Have Been Internalized by Cells. Please.
Thank you very much for your introduction. So I slightly changed the title of my talk because I have some new data I want to share with you. But the idea is the same. We're using, let's say, particles. We're doing delivery, and we're looking what is happening to the cargo. So let's say my group is focused on making particles, investigating the interaction of the particles with cells, and then at the end go to vivo. And I will be talking a little bit about this interface. Good. So there are a lot of deliverable systems, but there are certain problems associated with deliverable. One problem is that typically you want to deliver some molecular compound to the cytosol, but the problem is whenever you are working with particle-based systems, at the end you're ending up in the lysosome. So from this point of view, you have to find a way so that you can make the translocation from the things what you have delivered to the lysosome to the cytosol. And I want to show you several examples how you can do it with degradable materials and also how you can do it with remotely controlled materials. So the system I'm mainly going to talk about in the next time are polymer capsules. It's basically like a ping pong ball, and you have a shell around these capsules that's assembled by layer assembly, and you have a huge cavity, and in this cavity you can fill the molecules of interest. And the big concept what we want to make in some way some controlled delivery, because you want to get, let's say, the activation only if you've reached the target region and not outside. So the first thing we are trying to do is to work with, let's say, degradable prodrugs. For example, you can see here one molecule. This one is a protein, and this protein is overexpressed with fluorophores, so that the fluorescence is quenched. If you put this into such a capsule and you add it to a cell, everything goes to the lysosome, but then at the end it's stuck there. If you're working now with a biodegradable capsule, and this one is a material what can be digested by proteases in the lysosome, then you can see at one point you see the onset of fluorescence, and this one is due to the fact that enzymes can start chewing off the wall of this capsule. Then they even can cut this protein into peptide fragments, and now if these peptide fragments diffuse away, then it gets the onset of fluorescence. So you can see it's a compound what's non-active outside the cell, and only if it has entered the cell, then it's activated. And there's also some, so in this case, where it goes, it's not completely clear, because you can see here a fluorescence distributed along the cell, but we don't know in this case if it's really in the cytosol or if it's in exocytotic vesicles. But I will show you some other examples later. So now, this one is in principle possible. You just put something into a biodegradable polymer, and upon proteolytic digestion, something will be released to the cytosol. But how can you increase it? For example, for delivery of DNA. So what's commonly done in literature is using polyestyleneimine, because polyestyleneimine, it's a proton sponge, and if this one is inside the lysosome, it's basically changing the pH in the lysosome, and then there's some lysosomal swelling. The lysosome opens, and you get a release to the cytosol. So we tried now to make the same with our capsules. So we made the same as we did here before, but now inside, so the cargo was now DNA, but in addition, we added polyestyleneimine, and at the end you can really see the polyestyleneimine opens up the lysosome, it's distributed along the cytosol, and in this way you also get the expression of GFP. Or basically in this case, because it was siRNA against expression of GFP, if you're waiting for a longer time, you can see that this fluorescence is quenched. Good. But this one, it's now a more or less non-specific effect, but we wanted to see, can we make the release a little bit more specific? So we just can tune the geometry of these particles. For example, you can make these polymer shells out of more or less all materials, they only need to be highly charged. And DNA, for example, is a highly charged molecule, and you can bridge different strands of DNA by these linker strands. So if you assemble now the wall of these capsules, and that's work together with a group of Itamar Wilner, and you add, you use here a sequence, and the sequence forms an aptamer, then you can remotely control the assembly of this capsule. For example, in this case, this DNA sequence can form an aptamer with ATP. So that means if ATP is around, then the conformation of the DNA will change. It forms this complex, and in this way, this wall here breaks open. And if the wall breaks open, then you can get a release of this drug. What you can see here in this image. Good. So from this point of view, what I've shown you, you can use capsules that are, let's say, degraded either by some molecular trigger or just by some enzyme. But of course, there are still some limitation, and what at the end we would like to do, we would like to make a control what's completely done by the outside. And from this point of view, in order to, let's say, open up the capsule to create an artificial degradation, we're using plasmonic particles. So plasmonic particles, such as gold particles, act as local heat source. You can see here, this one is a cluster of a lot of gold nanoparticles, and you're moving in with a laser. Now this laser, if you are exciting at the plasmon resonance, the energy of this laser is absorbed. It goes first to the electron gas, from there to the crystal lattice, and from there to the environment. And you can see here, just by this energy, this cluster is driven apart. So now, what you can do, you can add these gold particles to the wall of this polymeric delivery vehicle. And now it's basically the same idea. You have this particle internalized by the cell, but nothing is happening because all the drugs are kept inside this polymeric vesicle. But now if you illuminate with a light pointer, you're heating these gold particles, and the wall of this capsule opens. And in this way, if the wall is opened, then these molecules can diffuse out. So if you're looking it into more detail, you're not only opening the wall of the capsules, but you're also locally perforating the lysosomal membrane. Because at the end, you get delivery to the cytosol, what you can do with some staining experiments. Of course, this one can be potentially toxic to the cell, but if you're doing it a controlled way, it's basically a transient poration, and the cell is tolerating this quite well. So in this case, it's an example. It's the thing what we want to deliver is mRNA for GFP. And how it's typically done, as I told you already before, you're either working with polyplexes, for example, with polyacylenamine, or you also can work with lipoplexes. And now if we add this lipoplex with mRNA to the cell, at one point you can see here the onset of the production of GFP. But the problem is, if you want to measure kinetics, so if you want to see how fast this process is, it's quite complicated because you have three time-dependent processes. The first one is that this polyplex has to be endocytosed. The second one is there has to be the translocation from the lysosome to the cytoplasm. And then you get the third process is the actual protein biosynthesis. So from this point of view, you can see here what you can plot. You can plot here the time of incubation, but you never know, for example, when one particle is inside a cell, because all these cells are individuals, all these polyplexes are individuals, so there's some kind of statistic distribution. If, on the other hand, you load this mRNA into a capsule, you can add this capsule to a cell. Then it's already in the lysosome. Now you open, and this opening defines now as a time point of 0. If you're opening, you know at this point you have the mRNA into the cytosol, and then you actually really can measure the kinetics of protein biosynthesis, because you have deconvoluted it from the uptake, because everything is already inside the cell, it's only packaged. But if you push the trigger button, then it's really released. And you can see here, for example, it's much steeper, the curve. And what you also can see, it levels at one point off, because here in this case, you're permanently delivering new polyplexes, whereas in that case, of course, you have only one shot. You deliver one time your mRNA, and then that's it. Good. So now we wanted to see if everything of this is also working in vitro, because what I've shown you so far is it was all based on cell cultures. If I'm talking now about in vivo, I'm not going to talk about mice or rats, because this one, of course, is possible, but there's one big problem, because if you want to do really afterwards an application, you always have the problem to bring the light as trigger into the tissue. Of course, we're working with near-infrared light, and so this one can penetrate tissue to some extent, but it's still in some way complicated. So we used as first model system, we used this animal, and it's called Hydra. It's a polyp. And this animal, it has a genome what's actually not so far away from the genome of humans, and it also has been highly studied in developmental biology. Because this animal is actually quite a quite funny organism. Let's say if you chop this animal here into 2 parts, out of 2 parts there will be the growth of a new animal. And how does it work? It works by molecular signal gradients. Let's say you have a signal gradient, and wherever the concentration of this molecule is high, you will get the tentacle, that's the formation of the head, and wherever the concentration is low, you will get the foot. So now you chop it apart here, the concentration here is lower than here, so it means here will be a new head. The concentration here is lower than here, that means here will be a new foot. So you really can just cut it into a lot of pieces, and by this, because this concentration of this signaling molecule is not the same, the animal will grow at one region a head and at the other region a foot. Good, so now the idea is just to package the signaling molecule, and in our case it's Alster-Paulone. So wherever there's Alster-Paulone, the animal thinks, okay, here's my head, and here I have to grow tentacles. The first thing what we can do, we can just package it again into these biodegradable capsules, and you can see here, this one is an animal, and if you add this growth factor to the animal, at one point the animal has localized these capsules all over the body, and now it thinks, okay, here is Alster-Paulone, therefore here's my head, and I have to grow tentacle. And therefore you got tentacle growth all over the place. But now, of course, that's not very controlled, and this one we can do now by this light pointer. So now we package the Alster-Paulone into the capsule, and nothing happens. But if we illuminate now, we again open this capsule, we release the Alster-Paulone. In this case, we can stimulate the growth of the tentacles. So I can see it here. These are the controls. It's without laser, without the gold particles, nothing happens. Just with the gold particles, but with no illumination, nothing happens. So there's basically almost no leaching. But if you have now the light-mediated release, you can see you get a lot of growth. So now at the end, what one still has to admit, because this animal is moving, we couldn't open just one capsule, what would be quite cool, because you can say, okay, here I illuminate, here will be your new heads, and you should get exactly one tentacle. So it's still not laterally resolved, but this one is mainly due to technical problems. But now let's go back again to what I said, because at the end, the problem is, of course, if you want to do it in a mouse, it's more complicated, because in a mouse, it's much harder to focus, let's say, light deep inside tissue. For example, in a human, if you go 10 centimeters to tissue, it's much more complicated. So at the end, I'm over the time. I just want to make one more statement. We can do exactly the same, but using, instead of gold particles, magnetic particles, and instead of light, using alternating magnetic fields. In this way, we also can create heat, and as magnetic fields are not absorbed by tissue, we also can do similar things in tissue. Good. Thank you very much.
Thank you. Thank you for a nice presentation. And I'd like to invite questions. So, okay.
Very nice talk, Wolfgang.
Thanks,
so you don't have leaching of the wind ligand in the 24 hours, right? So
yes, so I mean, leaching is always present, but in this case it was, let's say, below biological significance. And this one, I mean, at the end, I mean, you have to control by constructing the carrier vehicle. If you make, for example, the walls too thin, everything can leach out. If you make the walls too thick, at the end, you have a hard time to make the release, and you have to find an optimum.
Are you going to use this technique to do something with it?
I mean, that's at one point our plan. So what? So with let's say with the light, it's really complicated, because if you have a tumor deep inside the tissue, I think that will be technically really challenging. So what we are right now doing, we demonstrated that we also can open it with alternating magnetic fields. What you have, for example, in magnetic resonance imaging setup. Right now we are trying to make the release inside cells. So in cell culture, this one we have first promising results. And as long as this one works, I mean, we will go to a red, but it's nothing what I can promise, let's say, in the next year. But I think in the next year, we will have all the experiments on the cell culture finished, and then we can see it. But the good thing is for this magnetic resonance imaging, you have all the tools in hospital, so you can image, see basically where the particle is, and then you also could excite by magnetic heating.
And excuse me.
Don't worry. I'm more nervous.
No. It's fine. It's fine.
So, why don't we just take another question?
Thank you for interesting presentation. I wanted to ask you whether you know what are the efficiencies of the intracellular steps in delivery of the molecules, and what is the rate limiting step, basically? Going out of endosomes, or going to a nucleus, or whatever?
Good. So, I mean, this one is also a good question, because, I mean, let's say on a scientific level, it works, but there's definitely the efficiency. If we do this experiment, for example, on a cell culture, to open it is maybe 60 or 80%. The good thing, if we are doing it with this optical way, it's a single molecule experiment. So, I mean, the big danger is if you to open the capsule, it always works. But of course, if you open too much, if you heat too much, the cell is simply dead. So that's a good thing if you're working on a single cell basis. You put, for example, some viability test in parallel, and if you see, okay, something with the opening didn't work, you just go to the next cell. So in this way, basically, you get a success rate from 60 to 80%. But of course, afterwards, if you're doing it with in vivo, you would work on a multiple cell base, and then you cannot risk it. So let's say the limiting step from the application at one point is simply the homogeneity of the sample. Not all the particles have the same amount of, let's say, gold or magnetic particles, and in this way you need a different energy to open the individual capsules, and that's a technical problem. In principle it can be solved, but that's limiting.
Thank you. We need to, okay, just yeah.
Yes, this cell culture that you are testing these materials on are going to be three-dimensional or two-dimensional?
Sorry, if
I'm going to be three-dimensional cell cultures or two-dimensional?
So far, I mean, this one was all done for cell culture with two-dimensional, but I mean, the animal, of course, the hydra, so it's, let's say, it's millimeter size. So this one is really three-dimensional. And of course, I mean, I completely agree if you're working, let's say, for example, with this three-dimensional cell cultures, these particles are quite big. So, I mean, they will not be just penetrating to the, let's say, to the middle of this three-dimensional cell culture. I mean, this one is...
How big are the particles?
So in our case, for this, because we wanted to see them optically, we had them 2 to 3 micrometer, but in principle we also can scale them down to, let's say, 100 nanometer. If we go to a much lower size, we would lose all the advantage, because then the cavity is so small we cannot pack enough molecules inside them anymore. But 100, 200 nanometer, I mean, this one is no problem.
Thank you.
Good. Thank
you. Let's move on to the next presentation.